Introduction. Welcome to today's lecture on the intricacies of cell cycle regulation in biochemistry. In this comprehensive script, we will delve into the various aspects that govern the cell cycle, including cell signaling pathways, checkpoints, cyclins, CDKs, DNA damage response, and their significance in both normal and aberrant cell cycle regulation. By the end of this lecture, you will have a thorough understanding of the cellular mechanisms that control cell division and their importance in maintaining genome integrity. So, let's begin our exploration of the cell cycle. The cell cycle is a precisely orchestrated series of events that enables cells to grow, replicate their DNA, and divide into two daughter cells. It can be broadly divided into interphase and mitosis. Interphase consists of three distinct phases: G1, Gap1, S, synthesis, and G2, Gap2. During G1, the cell grows and prepares for DNA synthesis. In the subsequent S phase, DNA replication occurs, doubling the genetic material. G2 phase follows, allowing the cell to prepare for mitosis. Cell signaling pathways serve as the communication network within cells, relaying information and coordinating the progression of the cell cycle. Various signaling molecules, such as growth factors and cytokines, bind to their respective receptors and activate intracellular signaling cascades. These pathways regulate the expression and activity of proteins involved in cell cycle progression, thereby tightly controlling the timing and order of events in the cell cycle. CDKs are a family of protein kinases that play a crucial role in regulating the cell cycle. They are activated by binding to cyclins, which undergo periodic synthesis and degradation throughout the cell cycle. CDK cyclin complexes phosphorylate specific target proteins, orchestrating the events necessary for cell cycle progression. Activation and inactivation of CDKs are precisely regulated by a complex interplay of kinases, phosphatases, and cell cycle inhibitors. Checkpoints are control mechanisms that ensure the fidelity of the cell cycle by monitoring DNA integrity and repair before proceeding to the next phase. The major checkpoints are G1 checkpoint, G2 checkpoint, and the spindle assembly checkpoint, SAC, during mitosis. If DNA damage is detected or chromosomes are not properly aligned, these checkpoints induce cell cycle arrest, providing time for DNA repair or the correction of errors. Cyclins are a family of proteins that bind to and activate CDKs, promoting cell cycle progression. Different cyclins are expressed at specific stages of the cell cycle and collaborate with CDKs to regulate downstream targets. Examples include cyclin D, cyclin E, and cyclin A, which control G1/S and G2/M transitions respectively. Cyclin degradation marks transitions between cell cycle phases. Cell cycle inhibitors act as brakes, counteracting the activities of cyclin CDK complexes. They pause the cell cycle, allowing time for repairs, or prevent aberrant proliferation. Examples include P21, which inhibits G1/S and G2/M transitions, and P27, which halts the cell cycle during early G1 phase. The APC/C is a multi-subunit protein complex that regulates the transition from metaphase to anaphase during mitosis. It marks specific proteins, such as securin and cyclin B, for ubiquitination and subsequent degradation, leading to sister chromatid separation and exit from mitosis. The APC/C is tightly controlled and its dysregulation results in chromosome instability and mitotic defects. DNA damage response pathways are activated when DNA lesions occur during the cell cycle. These pathways involve signaling kinases, such as ATM and ATR, that trigger cell cycle arrests while DNA repair takes place. Failure to efficiently repair DNA damage can result in genetic mutations or aberrant cell division, potentially leading to cancer and other diseases. The G1 to S phase transition is a critical point where cells decide whether to enter the cell cycle or remain in a resting state called G0. This decision is controlled by the presence of growth factors, mitogens, and various signaling pathways. Activation of CDK complexes and the release of transcription factors enable entry into the S phase, initiating DNA replication. Mitotic entry and exit are tightly regulated processes mediated by multiple signaling pathways. Entry into mitosis is triggered by the activation of cyclin B CDK1 complexes and the phosphorylation of key substrates. Exit from mitosis is controlled by the inactivation of cyclin B CDK1 complexes and the degradation of cyclins, allowing for cell division and the restoration of interphase. Several signaling pathways function in concert to ensure the smooth progression of the cell cycle. Examples include the PEAK/ACT pathway, the MOPK/ERK pathway, and the WNT/beta-catenin pathway, each exerting specific control over cell cycle events. Dysregulation of these pathways can lead to uncontrolled cell proliferation and contribute to the development of cancer. Cancer arises from the accumulation of genetic mutations and aberrations in the normal mechanisms that govern cell cycle regulation. Defects in cell cycle checkpoints, dysregulation of cyclins, CDKs, and their inhibitors, as well as alterations in signaling pathways, can result in uncontrolled cell division and tumor formation. Understanding the molecular basis of aberrant cell cycle regulation provides crucial insights for developing targeted cancer therapies. In conclusion, the cell cycle is a tightly regulated process that ensures proper cell growth, division, and genome integrity. Its control involves intricate signaling pathways, cyclins, CDKs, checkpoints, and a delicate balance between promoting and inhibitory factors. Dysregulation of the cell cycle can lead to diseases like cancer. By comprehending the complexities of cell cycle regulation, we gain valuable insights into potential therapeutic strategies and interventions to mitigate the harmful consequences of aberrant cell division.